Hi, good morning, or indeed good afternoon or evening from wherever you're listening from. My name is Alex Sim, and I'm the president of AMSBIO. We've been around for 30 years, and we've gone from southern blotting all the way through PCR and next-gen sequencing. And now we find ourselves in this exciting space of organoid research, phenotypic readouts, and physiological relevance, which is really a pleasure to be here at the CMBL meeting that we've attended for a number of years. Thank you for spending some time with us. Today I'm going to give you a tour around some of the work carried out by most of our customers who are in the organoid field. We're going to cover stem cell derivation, cell culture, cryopreservation, the use of Lenti and Lentivirus, all the way through to a very unique high throughput microfluidic screening solution. Organoids have the potential to complement existing model systems and improve basic biology. Medical research drug discovery have improved immensely by this more physiological relevance. So one feature common to all organoids is they're generated from pluripotent stem cells or adult stem cells. And they can mimic the human development and organ regeneration in vitro, and they can do that for many, many passages and many long periods of time. So the analysis of organoid formation can therefore provide for us some very important information about the mechanism underlying human development and organ regeneration. And this is also helping molecular medicine. So it's also important because we can now get induced pluripotent stem cells or embryonic stem cells from different species. And this allows us to compare between the species, as one of our customers has done from EMBL in Barcelona. Mickey will be actually talking about her work. She is working importantly here with the pre-Semitic mesoderm cells. And she's used both our product for induced pluripotent stem cell culture, that's called StemFit, and her iMatrix 511 in her work. And rather important work it is because she's been able to, with the same conditions, work with mouse cells and human cells. Interestingly enough, she's using our silk produced iMatrix 511, which provides some benefits in terms of cost. The StemFit product also has been developed, as has iMatrix, because the importance of reprogramming to generate autologous cells for cell transplantation-based tissue repair is very, very important. And of course, if you're going to do CRISPR gene engineering, also can help, because then you can start to begin to transplant organoid type materials into patients. But of course, regenerative medicine needs animal-free product, and regenerative medicine needs products that are made to GMP. And this GMP has been worked on very carefully, and we now have a StemFit that is GMP available, and also the iMatrix 511 produced in CHO cells and both made in the GMP facilities. Of course, many people have been using iMatrix to replace, obviously, feeder cells, to replace Geltrex, BME, and replace Matrigel. The iMatrix is a fragment. 511 is a fragment of the recombinant laminin, and it does produce significant productivity gains. It's also actually very compatible with hydrogels, because one of the importances is to remove animal materials from GMP manufacture of cell therapy. And of course, the work of Matthias Lutolf, and in particular of Saba in his lab, has shown that their hydrogel can also be supplemented with laminin 511. Of course, once you've had all your organoids produced, at some point in time, you obviously have to freeze them down. You're building model systems, you need to come back to those model systems. Cryopreservation is very important, and I'm very glad to say that we have a wide range of cryopreservation materials under the Cell Banker name. One particular example is Cell Banker 1, and here you can see, for example, with these organoid airway buds, organoids, that you can see that the basal cells survive freezing. My colleague William actually spoke to someone in Hans's lab, to Dielte van der Vaart, And they caused the fact that they had to close down very rapidly on the closure, the lockdown period before COVID. And he was really pleased. And he says himself the procedure was a lot easier and faster, and he simply could transfer the organoids directly into Cell Banker and to freeze immediately. And that really genuinely helped him. So that was a nice comment from Yevte. I would say also that you don't necessarily have to freeze down in single test tubes. You can actually, and we have protocols of freezing down actually microtiter plates, whether it be 384 or 96 well. Other products within range include Cell Banker 2. Cell Banker 2 is a completely animal-free material, fully defined. You can see some references here from the Valier lab in Cambridge, UK. They were looking at cholangiosite organoids, and Noblik's lab as well has used our Cell Banker. I believe both of these people are attending and have posters and are talking at this particular meeting. The other important use of cryopreservation is also for stem cells. So you can freeze down stem cells in our stem cell banker. I would add that this is work showing here from Robert Arnes, who is actually Mary Hutch's lab. This work was done in Cambridge, UK. They were using the 3 stem cell bankers. The stem cell banker is a GMP product. It's available with DMSO and without DMSO. So it's completely available for cell therapies applications. And Mary Hutch, again, who's also talking at this meeting, is basically indicating here that day 2 and day 4 images clearly show that the organoids have recovered and have grown very well in cell banker freezing media. So three freezing media that you potentially can ask us for samples. We're quite happy to do that. Obviously, you want to be following regeneration, you want to be following development, and AMSBIO has an interesting range of lentivirus products. The awful lot here is showing long-term live imaging of epithelial organoids here using our cherry and our EGF stains and our lentiviruses in the organoids, and can follow the cellular dynamics and development. So a very, very good tool that can also shown here from Robert's work, again from the Mary Hutch lab, this time in Dresden, Germany, showing these cholangiocarcinoma nuclear red organoids, some fabulous photographs to follow progress using lentivirus. Lentivirus from the AMSBIO is a 3rd generation lentivirus. A quickly overview the facts from it. Replication incompetent, so they're very safe to use. We have a variety of products available off the shelf. We also have a high titer lentivirus available for animal work. It's important to note that the ready-to-made particles fluorescent proteins are great sellers, together with some of our reporter lentivirus for different genes, gene pathways, etc., such as Notch and Wnt, etc., important in organoid development. A lot of our customers are coming to us asking us for custom services. So they may have their favorite gene, and their favorite gene is either to be expressed or an overexpression experiment, or to be knocked down. We offer a guarantee 70% knockdown. We do the design for you. If not, you don't have to pay. Of course, with our overexpression, we can also build custom cell lines for you. If we move on to complement the lentivirus, we have adeno-associated virus. The advantages of adeno-associated virus include hard to transfect cells, and indeed we have a wide range of AAV serotypes that are used for such things as glia cells and neurocells, etc. Laying down your organoids, you probably need to have some ultra-low adhesion plates. Our Lipidure-Coat plates are available as flat bottom, round bottom, or V bottom. They can do aggregation of flat bottom multiple. Single U bottom are used for tumor spheroids in drug screening. And also single V bottom can be used for organoids, as is demonstrated by work here from Indiana, from the lab, inner ear organoids. And indeed, the plates have been used to produce some beautiful images here from Valerie in Professor Lacko's lab at the University of Newcastle in the UK. Working on these photoreceptors. The Lipidure-coated plates, she says, were extremely useful for producing homogeneous retinal organoids which responded to light. So you can see real phenotypic work working on the Lipidure plates, which are also available from AMSBIO. There's been a lot of work done on COVID-19 organoids are being used significantly to test for new drugs, etc., or to test for the uptake of the virus. And it's important, an antibody uniquely available from AMSBIO is a heparan sulfate antibody, which recognizes the heparan sulfate epitope that after digestion of tissue with the heparanase enzyme. And this has been used by Helena Meyer-Berg in the University of Oxford in her investigation of viral entry receptors in lung organoids. And we have a wide range of heparan sulfate antibodies, and of course of the heparanase enzymes, and of course heparan sulfate involvement in the extracellular matrix is very important, and I'm sure we're going to hear a lot more about heparan sulfate and heparin in the story of COVID. Finally, last but not least, we have developed a high throughput screening service. We can take your organoids or your spheroids, or even indeed patient biopsies, and make them into microtumors, and we can do co-culture cells, we can do combination studies, and we can look at in-drug efficacy. Tangible results are available from this. 20-fold increase in screening capabilities. We've got quantifiable tests of drug efficiency. We can take a biopsy, and in a real good clinically relevant window, we can produce some results using combination studies. You can choose label readouts of steroids. It works with perfusion, so there are no shear stress conditions, and certainly very much quicker results than using costly animal models. So this is a service. If anybody would like to speak to us, please come on to amsbio.com, or indeed go to Screen-in-3D, which is our new lab, a drug lab. Finally, just like to review what I've said to you. The StemFit cell media is available, both GMP and research grade. The iMatrix is available as recombinant, CHO expressed, or in silk. Our lentivirus solutions and custom services we've talked about, including also talking about our ultra-low adhesion plates called Lipidure. Cell banker freezing media. I hope it doesn't have to be used again in another lockdown, but if it's there, don't forget, it's very easy. Just drop your cells, your organoids, right into the cell banker product. Ask us for a sample, you can prove how it works. And finally, our screening services. Thank you very much for spending some time with us. I really do hope you enjoy the rest of the conference. Have a good day.